Hello. ഹലോ, ഈ സെമിനാർ എന്താ തുടങ്ങില്ലേ?
Okay, sir.
4:45.
4 more minutes, sir.
Sir, principal sir login ആകാൻ വെയിറ്റ് ചെയ്യാണ്.
അതെ, സമയം പാലിക്കണം. ഒരുപാട് ആൾക്കാർ ജോയിൻ ചെയ്ത് നിൽക്കുമല്ലേ. എനിക്ക് അമ്മ വിളിക്കാൻ പോണം.
Okay.
ആരെയും കാണാം.
Uh,
ഹലോ അനീസ, ഞാൻ ഓഡിബിൾ ആണല്ലോ അല്ലേ?
അതെ മിസ്സേ, കേൾക്കാം.
ഓക്കെ ഓക്കെ.
അനീസ സ്റ്റാർട്ട് ചെയ്യാം.
ഓക്കെ രജുഷ.
ഓംകാരം ബിന്ദു സംയുക്തം നിത്യം ധ്യായന്തി യോഗിനഃ കാമദം മോക്ഷദം ചൈവ ഓംകാരായ നമോ നമഃ. ഓംകാരായ നമോ നമഃ.
To one and all, thank you for sparing your time and being here today. We have gathered here today to attend the international webinar on conventional and unconventional T cells in health and disease, jointly organized by Department of Biosciences and IQAC Sri Shankara College, Kaladi. Before we move on to the welcome address, I would like to kindly remind all the participants who have joined us today to stay with us until the end of the session and access the feedback link for the certificate. Please ensure that your microphone is muted and video is turned off during the time of presentation. I now call upon the head of the Department of Biosciences, Srimati Jitasha Balan, to deliver the welcome address. Over to you, ma'am.
Thank you, Ashley. Good evening, everybody. A warm welcome to the respected dignitaries, faculty members, research scholars, and dear students. Today, we, the Department of Bioscience of Sri Shankara College, Kaladi, is conducting an international webinar on conventional and unconventional T cells in health and diseases to augment our
Person, Dr. Sherine, and all the participants. At the outset, let me congratulate the Department of Biosciences for organizing an international webinar on a very pertinent topic, conventional and unconventional T cells in health and diseases. We are all living in troubled times, times when COVID-19 has the entire world in its thrall. During these difficult times, it is very essential that we boost our immune system. This talk on T cells that helps protect the body against cancerous cells and cells infected by pathogens such as virus and bacteria will certainly be an enlightening one. I wish the webinar all success, and I hope that it will pave the way for some healthy interaction. Thank you all. Thank you.
Thank you, ma'am. Now I invite our beloved principal, Dr. A. Suresh, for the inaugural address.
സുരേഷ് സാറെ മ്യൂട്ട് ചെയ്തിരിക്കുകയാണേ. അൺമ്യൂട്ട് ചെയ്താലേ കേൾക്കുള്ളൂ. ഹലോ, സുരേഷ് സാറ് മ്യൂട്ട് ചെയ്തിരിക്കുക. ഹലോ, ഇപ്പൊ കേൾക്കാം. This subject is very relevant in the present scenario, particularly in light of COVID-19 pandemic, which has affected many countries across the world. There is a lot of research and studies going on T cells, as T cells are a key part of our immune system, and further studies of T cells will assist in diagnosis and treatment of many diseases. We have with us an eminent scholar and an expert in this area, Dr. Sherine Kottoor, King's College, London, who will be delivering today's talk. I express my gratitude to the organizers of this webinar for choosing this relevant topic, which I believe will benefit teachers, students, especially those doing research. I also take this opportunity to thank the management for all their support and motivation. With great pleasure, I officially inaugurate today's webinar and convey my best wishes for an effective, successful, and productive webinar. Thank you.
Thank you, sir. Now I invite Dr. Valsa A.K., Head of Postgraduate and Research Department of Microbiology, for the felicitation. Ma'am, please.
Good evening, all. I'm happy that the Department of Biosciences is organizing a webinar on conventional and non-conventional T cells in health and diseases. We all know that different types of T cells play a pivotal role in the maintenance of homeostasis in chronic inflammatory diseases, cancer, etc., etc. This lecture on T cells seems to be pertinent in the current scenario because one of the types of T cells called the memory T cells are thought to play a significant role in mounting immunological responses against several viral infections, including SARS-CoV-2. I'm sure that this talk will prove to be useful to all the teachers and students of biology. I wish this seminar all success. Thank you.
Thank you, ma'am. Our speaker for today is Dr. Sherine Hermangild Kottoor. I invite Dr. Anisa P.A., Assistant Professor of the Department of Biosciences, to introduce the resource person.
Thank you, Ashley. Thank you, Ashley. Of course, it's my pleasure to introduce you all our today's speaker, my friend, Dr. Sherine Hermangild Kottoor. Dr. Sherine hails from Pala, Kottayam. She has completed her Bachelor of Science in Biochemistry from the University of Kerala, Trivandrum, and her Master's in Biotechnology from the School of Biosciences, Mahatma Gandhi University, Kottayam. She received the prestigious Marie Curie Research Fellowship from the European Union and was awarded her PhD for the thesis entitled Regulatory T Cells in Human Uveitis from the University of Birmingham. She has many achievements and research experiences added to her credit. Her journey in biological research from IIT Bangalore as a trainee. She worked as a summer research fellow at the Center for DNA Fingerprinting and Diagnostics CDFP, Hyderabad, India. She was a visiting research scholar at the Institute of Genetics, University of Notre Dame, UK, and Columbia University, New York, for a period of 3 years on prestigious projects in the field of developmental genetics and cancer immunology. She was also an exchange research visitor at the Department of Ophthalmology, Ludwig Maximilian University, Munich, where she worked on animal models of ocular diseases. She worked as research associate at the TB Research Center, Imperial College London, under a project entitled Early Detection of Latent Tuberculosis. To her credit, there are lots of international publications in peer-reviewed journals with high impact and good number of citations. She was also an active participant in a large number of international congresses hosted worldwide. By profession, her expertise is in T cell immunology. Currently, she is working as research associate at the School of Immunology and Microbial Sciences, King's College, London. Today, she is with us to present a talk on conventional and unconventional T cells in health and disease. Thank you all for joining us. Now, over to you, Ashley.
Thank you, ma'am. It's time to start our technical session. Before we begin, let me explain the arrangement of the session. The session will begin with the presentation of the speaker, followed by a question and answer session at the end. For raising your queries, you need to put the questions in the chat box, which will be answered by the presenter during the Q&A session. Participants, kindly make sure that your audio is mute and video is turned off to avoid any disturbances during the presentation. Dr. Sherine, the platform is all yours. Let's begin the presentation.
Thank you all. Good afternoon, everyone. This is still good morning here. So first of all, very thank you to all of you for inviting me, especially Honorable Principal Dr. A. Suresh and the Head of the Department, Mrs. Jitisha Balan, and all the dignitaries there for inviting me and giving me this platform to share my work with you. Before I start, I wanted to raise a problem because I can't share my screen with you for some reason. Is that a problem? Because I'm not able to share the screen, share my presentation with you.
Yeah, because you are not in the presenter mode. I think somebody needs to put her in the presenters list.
Yes, please.
Anisha, please change your status to presenter.
Sherine, just hold on.
Yeah, no worries.
Yeah. Sherine, just hold on for a minute.
No problem.
Okay. Hi, Dr. Sherine. Could you please join us through Microsoft ID? This is provided to you.
Okay. Let me log out and log in again.
Okay.
Hello. Sorry about the confusion.
Okay. Can you share now?
Yes, yeah, I hope I can. Can you
Yeah.
all see?
Yeah.
Okay,
Yeah, it's
good.
fine.
All right. So today I thought I would give you a flavor of what we had been doing in the immunology field here, especially the work I had been doing for the last 3 years. So at least I hope for the students that you will get to know, get an idea of what all the theory you learned from the textbooks, how they are being applied in the research field. So my work for the last 3 years had been in inflammatory bowel disorder, which is an autoimmune disease. So inflammatory bowel disorder, or IBD, they are a group of inflammatory conditions that can affect the colon and the intestine. There are 2 different types of IBDs. One is called ulcerative colitis, the other is called Crohn's. Basically, ulcerative colitis affects just the ascending part of the colon here, whereas the Crohn's is more extensive and it can affect any part of the colon. So this gross appearance is very characteristic of the colitis. So you can, as you can see, this is the intestine from a deceased patient, and the mucosa is quite eroded. So this is a very much the characteristic of this colon from these patients, and these patients come with anemia, skin rashes, lots of bloody diarrhea, etc. These are some of the endoscopic pictures from these patients we see, and you can see there's quite a lot of ulcer, the mucosa erosion in these pictures. The main causes of IBD is still unknown. As I said, it is an autoimmune disease. So whenever I say IBD in this presentation, you can easily replace it as autoimmune disease because most of the mechanisms are quite the characteristics of many autoimmune diseases, such as rheumatoid arthritis or MS or systemic lupus erythematosus. So it is mostly caused, there are genetic loci with polymorphism that has been identified that can cause IBD. There are environmental factors which are coming up in recent years that can cause, especially the microbiome. And it's quite interesting that IBD was supposed to be a Western disease. India or Asia, in Asian continent, we didn't have that much IBD to begin with. But with the change in dietary habits and the environmental effects, you can now see lots of IBD cases coming up in the Asian continent. So environmental factors do play a major role in IBD pathogenesis. And most important of all, there is that dysregulated host immunity, which is the part I am more interested in. You might all have learned this already in your immunology course about the Cd4 T cells. So Cd4 T cells are believed to be a major player in the IBD pathogenesis, mainly because in the gut of an IBD patient, you can see an accumulation of these CD4 positive T cells. And it is the same in a rheumatoid arthritis joint or a multiple sclerosis brain. So these CD4 T cells accumulate at the site of inflammation in most of the time. So this is just basics, as you would know, that naive CD4s, they haven't seen any antigens. Once they see the antigen, they get activated and they become memory cells. So there are 2 types of memory. One is protective memory, which are the effector memory T cells. So one of the major characteristics of the naive T cell is that they express this molecule on their surface, CD45RA. Once they're activated, the RA turns into RO, CD45RO, which is the characteristics of a memory T cells. So the effector memory T cells mediates the protective memory. So they can migrate to inflamed tissues in the periphery and display immediate effector functions. Whereas these are the central memory T cells. they are the reactive memory cells. So they can't go into the tissues, but they can home to the secondary lymphoid organs where there are other T cells, and they don't have any effector function, but they can readily proliferate and differentiate into effector cells in response to an antigenic stimulation. So these are the major, the memory cells are the major players in any form of inflammation. You might have learned about the different type of T helper cells, or because CD4 cells are called helper T cells because they help other cells in their function. So this is a very basic diagram of the different T helper cell subsets. So you can see the naive cells, they receive signals from the cytokines in their environment, which is listed outside the circle. Recognizing these cytokines, they get activated. This in turn, this activation lead to distinct signaling pathways, which is in the outside circle. You might have heard some of these names, at least STAT4 and STAT5. This in turn leads to the activation of different transcription factors, which drives the expression of various cytokines that defines different T helper subsets. For example, Th1 cells express Tbet, and they produce the cytokines interferon gamma and TNF alpha. Th2 cells express GATA3, and they produce cytokines such as IL4 and IL13. Th17 cells express ROR gamma T as their transcription factor, and they produce cytokines such as IL17 and IL22. And there are these regulatory T cells, which they express a specific transcription factor known as FOXP3, and they produce cytokines such as IL10. Their main function is to regulate the whole immune reaction. Also, before I go on, if anyone has any doubt on, or if I'm going very fast, please stop me and ask me any questions during the presentation. I wouldn't mind. So in the recent years, there have been a lot of studies on these different T helper cells, how to best define them, how to easily define them. So you would have learned about chemokines and chemokine receptors, which help them to move to specific sites. So specific T helper subsets also express specific chemokine receptors on their cell surface. I'm going very fast on this bit because I think you all would have learned these in your textbooks already. So these are the major CD4 positive T cells that play, that are the major cellular players in IBD. So we have Th1s, which has CXCR3 as the chemokine receptor on their surface, Th17s, which has CCR6. And there is this recent hybrid cells. They have the characteristics of both Th1s and Th17 cells. They express both sets of transcription factors and chemokine receptors, and they also produce both sets of cytokines. They are supposed to be a very potent pro-inflammatory cytokine, which causes lots of inflammation in various disease sites. Also, we have the regulatory T cells. So in a normal circumstance, this is how the body balances itself. You have the pro-inflammatory bits, but you also have the anti-inflammatory cells and their signaling pathways to counteract the pro-inflammatory bits. But in an autoimmune disease like IBD, there is not just one bad player. There are lots of pro-inflammatory cytokines, cells, environmental factors, everything plays a role, and they all form one big bad guy. For example, if you have Th17s, which is normally okay, but in an inflammatory environment, the Th17s produce lots of IL-17s, which can recruit monocytes and neutrophils to the site, which can then activate other downstream cytokine cascade like TNF, IL-6, and other chemokines, thereby making it difficult for the regulatory cells to regulate the inflammation. So in this study, I wanted to look at the different type of T helper subset playing a role in the IBD pathogenesis. First, I wanted to look at healthy individuals to see how, in a normal individual, these players are there. So we took blood from both patients and healthy individuals. Then we isolate the mononuclear cells with a density gradient centrifugation. So all the mononuclear cells will be at this juncture, and this will have all the lymphocytes in there. So the lymphocytes are then stained with antibodies, which has fluorescent labels attached to those antibodies. So, and then these cells are passed through this machine, which is the flow cytometry machine. So flow cytometry is a technique whereby you can stain multiple cells with different types of antibodies. So you can, from one sample, you can identify almost all the cells in that sample. So this is how a flow cytometer works. So you have the fluidics inside the machine move cells one at a time through this laser. The light scattered by the cell, which is this blue light, it gives us an idea of the size and complexity of the cell. The bigger the size, the bigger the scatter will be. Don't worry, it will come on. And also, the fluorescent molecules that are bound to the cells are also excited by the laser, and they emit light at a particular wavelength, which are then detected by various filters and detectors. And the machine then quantifies and gives us an output. That is how we identify different cells. So different fluorochromes, but these are the fluorescently labeled markers, will give out specific signals at specific wavelengths. And the machine gives us an output in voltages. So this is how the cells would look like when it comes out. So these are the cells that express all the CD8 antibody, CD8 marker, and these are all the CD4 cells. So we take the CD4, because I was interested in the CD4 cells, I took the CD4 cells and looked at the expression of all the other markers that can define the various T helper cells. So from the CD4 cells, you look at the regulatory T cells and the normal conventional T cells. The conventional T cells, then again, we look for the other markers. There are naive T cells, which express CD45RA. We have the memory T cells, which express then the CD45RA, then becomes CD45RO. The memory T cells then divide into effector memory and central memory. So the effector memory cells are the ones that then differentiate into the different T helper cells. And based on the chemokine receptor expression, we then divide them into Th1, Th17s, Th117, and Th2s. Again, I will tell you, if I'm going too fast, please stop me. And so you might have read about regulatory T cells, but in recent years, there had been lots of research going on regulatory T cells itself. And in 2012 or 13, there has been this new development in the regulatory T cell field, showing that Tregs are also, these regulatory cells can also be divided into these type of T helper like cells. So even within the regulatory T cells, we have the naives and the memories or the antigen experienced Tregs. And they can then be divided into the Th1-like Tregs and Th17-like Tregs. So nobody quite know why we have these Th1-like Tregs or Th17-like Tregs are there in the body. And most of the consensus is that it helps them to co-localize with and effectively regulate the different types of T helper cells response in vivo. So this is from a paper we published this year. So this figure B shows a different way of analyzing these cells. This is by looking at the cytokines they produce. For example, so if you look at the CD4 cells, those that express IL-17s are the Th17 cells. Those that express interferon gamma are the Th1 cells. And those that produce both gamma and IL-17 are the Th117 cells. So to look at these cells in a healthy state, we first recruited healthy controls, healthy samples, healthy individuals. So we had another study going on, which was on monozygotic twins. So we recruited 100 pairs of monozygotic twins. This is very important because these are monozygotic. That means they are genetically identical. So these twins are genetically identical. The only difference they have are the environmental factors. Some of them are quite old. They live in even different continents or different places. They live in different households. They have different dietary habits. But what was most important was, even with all those environmental differences, you can see if you compare one twin to the other, the frequency of the different T helper subsets remains quite identical. For example, if one had a high Th1 cell content, the other one also had, except for a very few non-concordance twins there. So this was a very, very interesting find, and this was published this year. So one of the reasons we looked at twins is mainly you would have heard about microbiome and metabolome mostly, which is quite a trend now. It's mainly because, why is microbiome important? Because it had, especially why is microbiome important in immunology? Because there is accumulating evidence that show that presence or absence of particular species of bacteria or fungi inhibiting or colonizing the host gastrointestinal tract can actually regulate the generation of different effector and regulatory T cell subsets. So these are all the work done in mice, and we are actually doing that in humans now. So for example, so this bacteria, segmentus filamentous bacteria and Clostridium, has been shown to induce the induction of Th17 cells, whereas Bacteroides fragilis could induce Treg cells. So that is why we were looking at the twins, because they are genetically identical. So any difference we see could be due to their microbiome or metabolome. So we are in the process of looking at the microbiome from these patients and also the metabolite of this bacteria. I can't show you the results yet. Anyway, so back to the IBD research. So as I told you, there is quite a lot of accumulation of these CD4 positive T cells in the gut in these patients. So how do those cells get into the gut? So this is called homing of T cells. You would have heard the word, but how do they home to that particular site from the circulation? So T cells, when they are activated near the lymph nodes surrounding the gut, where they encounter gut-specific antigen, they express this particular molecule on their surface called alpha-4 beta-7 integrin. So this molecule, when the T cells express this molecule, its ligand is this molecule, MAdCAM1. So this ligand is expressed on the gastrointestinal tract. So once these cells express these molecules, they can then home to the gastrointestinal lining where they bind to MAdCAM1, which then helps the egress of the circulating T cells into the gut. So in my study, I wanted to look not just the whole lymphocyte population, but I also wanted to know the cells that are destined to go to the gut by expressing the alpha-4 beta-7 integrin. So those cells we call gut-homing T cells. So this is the first diagram, again, just to show you the gating. Okay, So these are the results. So we first looked at the different T helper cell subsets from so healthy control versus ulcerative colitis. As you can see there, between the healthy controls and the colitis patients, you don't see much difference in the frequency of cells in T helper 1, 2 cells. But there is quite a higher proportion of Th17s in the blood in UC patients. There was no difference in the regulatory T cell compartment. But remember what I said in the beginning, that in an inflammatory condition, you get all these cells. You also get regulatory T cells into the gut. So it's not the actual number of cells that matters. It is actually the balance between the regulation and the inflammation that matters in the body in a biological context. So we looked at the balance between the regulatory T cell and these pro-inflammatory T helper 17 cells. And as you can, as you would hope that in UC patients, that balance is quite low. There is less Tregs to Th17 ratio. So anyway, as I said, there are different even within the Tregs, there is different subsets. So I wanted to see how that balance is. So if you look at, so this is the healthy control. If you look at the Th1 cells compared to Th1-like Tregs, there is quite a good correlation going on in healthy control. This is the same in all such subsets. So Th17 to Th17-like Tregs. So the more Th17s you have, the more Th17-like Tregs you have. So that's how they tightly control the inflammation in a healthy control. But when it comes to the when it comes to the UC, ulcerative colitis patients, this balance is not there, especially in the Th17s. Th1s and 117s are okay, but it's the Th17s, you lose that correlation, showing that that balance is disrupted in these patients. Then we also looked at, as I said, the gut homing cells, which express, because these are the ones that would eventually go into the gut and cause the inflammation there. So we looked, so this is how a flow cytometry plot, this is the expression of alpha-4 beta-7 in different cell populations. These are the beta-7 positive cells, and these are the negative cells. As you can see, there is, within the Th17 cells, again, the gut homing Th17s were higher. And I know this is a very busy slide, but the take-home message is that the disruption we saw in the Treg to Th17 ratio is still there in the gut homing compartment, which is quite important, especially in the context of an IBD patient. So one of the ways we treat IBD, so there are different treatment options for an IBD patients. So some of them are by inhibiting specific pathways or inhibiting specific cytokines. So one of the very common treatment is inhibiting TNF. So anti-TNF antibody is commonly used for rheumatoid arthritis and IBD and various autoimmune disease. So by blocking the anti-TNF, which is a very high pro-inflammatory cytokine, you expect to reduce the inflammation. But it has the problem with that kind of approach is TNF is produced by many different cells, and it can cause lots of side effects, and people get desensitized. They lose their response to the treatment over time. So one of the new advances in IBD treatment is an antibody called Vedolizumab, which actually binds to this alpha-4 beta-7 integrin. What that does is it effectively blocks the cells from entering into the gut because the alpha-4 beta-7 can't bind to its ligand at the intestinal tract. So This is quite an effective treatment. However, we don't know till date how it affects the different T helper subsets. Of course, we wanted to block all the pro-inflammatory cells from going into the gut, but we also don't want it to block all the regulatory T cells. So we looked at the patients getting this treatment. So we got samples from them before treatment, that is 0.0. Then at different time points during their treatment, and then we looked how their T helpers compartments change over time. So this black line is the vedolizumab, and this blue line is our control. So in any experiment you do, to have a control is very important. So an experiment control. So this is the control here is the anti-TNF that I just mentioned. We compared the effect of these two drugs on the different T helper subset population from these patients. And it's quite evident from the first graph that it doesn't affect the Th1 or Th2s that much, because sometimes it goes up, but it always comes back to that baseline frequency. But the Th17s and the hybrid 117 cells are the ones that go up in the blood, which is what we want, because as we progress, the more we block these cells from going into the gut, there will be more of these cells in the blood, but that's okay. We have other machineries. It will go into apoptosis soon after. The Treg subset is a bit different. You can see there is a bit of blocking of Tregs, but not as much as the Th17s. And this is how this is why the treatment works, because it's effectively blocking Th17s. But the problem is, even though this treatment is quite effective, it's only effective in about 50 to 60%. So we wanted to see what's different between the responders and non-responders. What is the immunological difference between a person who responds to a treatment and compared to the person who doesn't respond to the same treatment? So we looked at the patients who receives this treatment, divide them into responders, which is the dark black line, and non-responders, which is the dotted red line, and track them over time to see what is actually happening in their immune system over time. What is the difference between responders and non-responders? And again, so you can see here, this is the major difference, the Th17s. The responders has the ability to block more Th17s in their system, whereas non-responders, they don't block them as effectively as the responders. So now, if you look at any medical journal these days, you will hear about precision medicine. So what is precision medicine? Is to this is tailor-made medicine. So tailoring the treatment to each patient. Because this is a lengthy treatment, it's always scientists are always looking to see if there is a chance for us to identify people who will respond to a specific treatment even before they start the treatment. That's the best case scenario. So if we could identify people who would respond well to specific treatment, and who would not respond, then there is no point in treating them. So we looked at the baseline characteristics of these patients to see if we could find a biomarker to actually point out response. And this is what we found. We found that a combination of two immunological signatures, which is decreased Th17s and increased Tregs at baseline, could predict vedolizumab response. So this is something we we are planning to apply in our clinical setting soon to see if that would be helpful to figure out the treatment response. So that's this is one of the papers that we are going to publish in the next. We're sending to publisher in the next two weeks. Then, as I said, I also said unconventional T cells in the title. So these are the conventional cells, but there has been a lot of interest in the field of unconventional T cells. So what are they? So you have learned about the T cell receptor rearrangements and different T cell receptors, alpha, beta, gamma, delta, etc. But there is another group of cells called mate cells or mucosal associated invariant T cells. There are two sets. So these have a different type of TCR basically, and they are, you would have learned about MHC class 1 and class 2 molecules and how they are required for the for antigen presentation for the normal conventional T cells. So these are unconventional cells. So they are not restricted to the MHC classes. They have a different set of molecules they are restricted to. These mate cells are interesting because they are found quite a lot in the intestinal side. That's why they got that name, because our colon is one of the major mucosal site. So they are present in the lungs, all the mucosal side, lungs, intestine, and liver, which is why these cells are quite now studied in the COVID response, because there's quite a lot of mucosal associated mate cells in the lungs. Another interesting factor is they can produce all the cytokines, almost all the cytokines. They produce interferon gamma, they produce IL-17s, they even produce granzyme, which is a killing cytokine. So even now, nobody knows whether they are a friend or enemy of the immune system, because it has been shown in various autoimmune and immunological disease. In some disease, it has been shown to have a regulatory effect, whereas in some, they seem to have a pathological effect. I'll just show you a bit of what we are planning to do. We are still recruiting patients for this experiment. So we wanted to check different, not just the conventional T cells, but other unconventional cells that are involved, not just in blood, but also in the gut of these patients. So again, we use flow cytometry, but the problem is we want to look at the cytokine expression. For that, this is, I don't know whether you have learned it. So this is the normal, most used way of cytokine staining for flow cytometry. Because we take out the cells from the blood, and you need to stimulate these cells with an antigen for them to produce the cytokines. But normally what happens is when the cells are stimulated, they produce the cytokines within, and their Golgi complex would take the cytokines and secrete it to the outside. But we don't want them to be, we want them still inside the cells, because otherwise we wouldn't know which cell it came from. So we stimulate the cells, and at the same time, we give them a protein transport inhibitor, which stops the Golgi from transporting the cytokine to the outside. Once they are stimulated and the Golgi is stopped, you do the normal staining for the surface molecules like CD4, CD8, CD45RA, RO, CCR7. Then you fix the cells so that all the surface antibodies stay on surface. Then you poke little holes in the cells and you stain them with the cytokine or the intracellular antibodies for their detection and do the flow cytometry. So this is one of the biggest flow cytometry panel we have done, which is 16 colors. I think that is the maximum you can do on any flow cytometry machine we can do, but that is available now. So here we can identify CD3 positive cells, which are all the lymphocytes. We have CD3 negatives, or then you can also look at the NK cell population in there. We have the invariant different T cell receptor antibodies to look at the gamma deltas, the mates, and the iNKTs. We also can look at the normal conventional CD4, CD8s, and also the various various cytokines. As you will look at, if you look at any of the Nature or PNAS or Nature Medicine papers, you will see graphs like this, plots like this. So this is another way of plotting your flow cytometry data. Editors nowadays are very stringent about how you would show or analyze your data, because when you're analyzing your data, there is always a receiver or operator bias. You want your patients to be there or healthy controls to be there, even though you are not consciously looking, there is some unconscious bias. So this is an unsupervised analysis. You take your raw data from this flow cytometer and put it into an algorithm, and it is the computer that then clusters different cells based on their markers. So it's not up to you, but it's the algorithm that will treat every single sample the same way. So you can see, especially with this kind of big panels and big data, this is one of the ways they show data these days. So you can see the different cell subset within the blood and gut from the patient. So this is blood before the treatment. You have CD8s in red, we have CD4s in brown, mates in purple, a little bit of gamma deltas here, and very few iNKT cells. And you can see the cells changing over time before and after treatment. And you can also see which cells express which cytokine. Some you can see in blood, at least most of the IL-17s, mainly from CD4s and CD8s, and the granzyme mostly from CD8s. So this is one of the ways we do our analysis. Now, before stopping, I would like to show you why these kinds of flow cytometry panels, when in a pandemic like this, how do scientists produce data so fast? Once you have the infrastructure and the techniques in hand, you can apply this to any disease, any human disease in any conditions. So this is one of the papers from one of my colleagues, some of my colleagues in King's, and they have actually, when they started this, they have actually used my panel using the CXCR3 to address the different T cell population, to look at the various immune signature from the patients. This is a latest Nature Medicine paper. And this is another way people look at the different cells. So this group has taken cells from the patients and the healthy controls and then stimulated them outside in the lab, in vitro stimulation, using COVID antigen, and look at which cells are stimulated. And you can see in COVID patients, the gamma producing Th1 type cells and the IL-17 producing Th17 type cells are more excited, meaning that they are more involved in this. So these are some of the recent reports on T cells in the SARS-CoV-2 infection. Lymphopenia, especially in the T cell numbers, has been widely reported in COVID patients. And from one of the papers from Wuhan patients, they have showed that the naive cells in the periphery are increased, whereas the memory cells had been decreased in the periphery in severe cases. And scientists think this is because of that trafficking of these memory cells to the site of inflammation, which is the lungs. So there is lots of research going on in this area, looking at the trafficking of cells at the moment. Then they have looked at the various pro-inflammatory cytokines that are involved that are produced in the lungs, and they have shown that these pro-inflammatory cytokines such as gamma and IL-6 and some of the chemokines are increased in the lungs, which could then attract the immune cells like T cells from the peripheral blood. And again, as I said, there is that balance. It's most important in the immune homeostasis, because the IL-6 cytokine is very interesting because it can, in the presence of TGF-beta, induce the production of Th17 even, and block Treg production. So thereby contributing that dysregulated balance between Tregs and Th17s. So I think that's my presentation. I hope I gave you a flavor of at least what the practical side of the things you learn in theory is. And once again, thank you all for listening, and thank you for giving me this platform. And a very big congratulations to the coordinators who organized this kind of event, because this is such a very strong platform to bring international research to your doorstep. Thank you. I'm open to any questions now.
Thank you, ma'am, for your very informative and scholarly presentation. Now it's time for the interactive session, and I request the participants to come forward and put your queries in the chat box in order to join our discussion.
Sherine?
Yes.
Okay. Could you add something about the relevance of the T cell involved pathways, especially in case of pandemic, situations?
Right. So again, as I said, it's not just one cell. It's always the pathways. That's why in any disease research nowadays, we just don't do the normal just a T cell analysis. We do single cell RNA sequencing and microbiome and metabolite to see the pathways involved in, especially the. For example, the IBD, the JAK-STAT pathway it's very important, and there is actually a treatment that is being developed in collaboration with our lab, with a pharmaceutical company. So I mean, even in COVID, these JAK-STAT pathways, they are still looking at the pathways involved in the pathogenesis, but it's still early days now. Okay, so how far we could apply facts in large-scale diagnosis of COVID-19 in the Indian scenario. So again, as a diagnostic tool, I don't think the facts has been developed yet as a diagnostic tool, because it's still the PCR, because we are looking at the viral load rather than the cells involved. So even here in UK, we don't use facts. It's mostly for the research purpose that we use facts for. But I think because it's still in the early days, we still don't know which cells are involved, which pathways are involved. So I think it's still early days for that. Okay, Vedalisima. So what is Vedalisima? So Vedalisima, as I explained, there's alpha-4 beta-7 integrin, which appears on the cell surface when they are activated. So, and that helps them to traffic to the gut. So vedalisumab is an antibody developed which can bind to this alpha-4 beta-7 integrin on the T cells. By binding to it, the T cells can't then go and bind to its ligand, which is expressed on the gut lining. So because this interaction between the beta-7 integrin and the ligand is blocked, the T cells can't then enter from the blood, cannot enter into the gut and add to the inflammation there. So that's how we find it even in the patients, if you look endoscopically, because I assist the endoscopy, and you can actually see the difference in the number of cells in those patients from the histology. I hope that's okay. Clear?
Ma'am, we've got one more question. CD45 is a phosphatase enzyme. What's the difference between CD45RA and CD45RO?
So that is different, actually. CD45, again, the chemistry of it, I am still weak at it, but CD45 is different to CD45RA and RO, actually. So CD45 is actually all lymphocytes expressed, irrespective of a T cell or a B cell. They all express CD45. But CD45RA is mostly related to the naive, the antigen experience of the cell. So they are both completely different molecules, actually.
Okay, ma'am, thank you so much. I believe that Dr. Sherine has answered and covered all the queries raised by the participants. Ma'am, would you like to share anything else?
No, I think I'm deeply grateful for this opportunity. It's wonderful to share my work with someone in India, especially. So, and I wish the organizers of this webinar series all the luck, all the best wishes for your future webinars, and best wishes to all the students. Thank you.
Okay. Thank you, ma'am. It's time to wind up our session. I invite Srimati of the Department of Biosciences to conclude the session with a few words on the talk.
Good evening. Honorable Principal Dr. A. Suresh, IQAC Coordinator Dr. Preeti Nair, Resource Person of the session Dr. Sherine, HOD of Postgraduate and Research Department of Microbiology Dr. A.K. Valsa, Mrs. Jitasha Balan, HOD Department of Biosciences, my dear colleagues, students, and all the participants. I feel privileged to have been asked to propose a vote of thanks on this occasion. I, on behalf of the Biosciences Department, would like to extend our hearty gratitude to the honorable delegates who blessed us with their presence and took out valuable time of their busy schedule for this event. I take this opportunity to express my sincere gratitude to our principal, Dr. A. Suresh, for the encouragement and strong support rendered in the successful conduct of the webinar. I would also like to thank the IQAC coordinator, Dr. Preeti Nayak, for her wholehearted support to organize this event. I would like to convey our deep appreciation to Dr. Sherine for her talk, which was very informative and interesting. Hope that all of you had a fruitful discussion in the Q&A session. The webinar titled Conventional and Unconventional T Cells in Health and Diseases was mainly focused on T cells involved in autoimmune diseases, particularly inflammatory bowel disease. It was really interesting for all of us to know about the immunotherapeutic potential of T cells. Thank you once again, Dr. Sherine, for the well-structured and informative presentation. Hope it really enlightened the audience. I'm sure that the content of the webinar is indeed an inspiration for. Next, I would like to extend my sincere thanks to Sri S. Prasad, Assistant Professor, Sri Shankara College, Kaladi, for the technical support right from the beginning of the webinar. I would also like to thank all the participants for their positive interaction in the webinar. Just like any successful event, this too would have been impossible without the active involvement and support of all my colleagues at the department. I thank them all for the great support and help. Thank you all once again for making this event a grand success. Thank you all.
Thank you, ma'am. Dear participants, please note that the feedback link is provided in the chat box. Kindly note down and fill it soon. We are also taking this opportunity to announce that our next international webinar series, Biogen 2020, will commence from September 7th onwards. Hope you all will join. Once again, I thank everyone for being here. We appreciate your kind cooperation. We will see you next time. I request all the participants to fill up the feedback form before leaving. This link will be active only for 1 hour after the session. The e-certificates of the webinar will be issued to the respective registered email address of the participants within 3 to 5 days. Let's disperse for now. We will see you next time in our international webinar series, Biogen 2020, on 7th September. Thank you and have a nice day.